Our next talk is by John Witman, who's going to continue our exploration into the marine environment.
Well, thanks for that detailed introduction, Eric. So I'm going to continue on some of the themes that Julia so nicely brought up. She talks about a snapshot in her 4,000-year record. I guess our 20-year record is more like a screenshot lasting about a second. But you'll notice that the title of our talk changed a little bit to features, drivers, and futures. Not stock market futures, but ecological future research questions, which are a lot easier to predict, I'm sure. And we work on the seafloor, as you can see from that great diagram from Van Loon's geography. Probably a pretty good depiction of what Galapagos is really like. And we work on those communities over the other side of charismatic benthic invertebrates, such as gorgonians and sponges and, yes, corals, and the reef fish and marine mammals and large megafauna that interact with these communities. So we're very much kind of a whole community group. And also note that I added Robert Lamb as a co-author. Robbie is here in the audience, and he did a PhD with me, or survived one, in the Galapagos. So this is the year of the World Cup. So I was thinking how to organize this talk, and I decided to use the soccer star cards, which are traded like baseball cards. Here we have 3 stellar stars of soccer from South America, and instead of soccer superstars, I decided to organize it by ecosystem stars of the Galapagos. And yes, this was a late night idea, and I got carried away, and I printed out these cards. There are 5 of them, and they're available at the coffee bench. And among other things, they make great bookmarks. So anyway, Galapagos All-Star #1 is the steep bathymetry. This is not anything new. The geologists have known this for a long time, but it's enormously significant in understanding the marine ecosystem, because what you might see from this bathymetric map that's in one of our papers, sorry for making you seasick, that's not going to work. Can I use this as a mic?
Yeah.
Okay, so look at this corner of Santa Cruz. The bathymetry, it's very steep. The isobaths are really close together. It's basically like diving on the edge of the Grand Canyon. It goes from the shallow marine ecosystem to the deep sea within a very, very short distance, which you can see here in Stuart Banks' 3D rendition of that bathymetry. Baltra, our study sites along this region are Baltra and Racas Gordon. And these are strong upwelling sites, also not so surprisingly. And one of the ecological consequences of the steep bathymetry is we have the occurrence of species that normally live in the mesophotic zone, that's below 30 meters, and in the deep sea, living at very shallow depths in the Galapagos. And I doubt you can read the text, it's not important, but this is one of these species. Black corals, we probably have documented the shallowest global distribution of black corals anywhere. They live up at 3.4 meters depth and down to hundreds of meters. And they're ecologically important because they're what we call foundation species that provide the architecture, the habitat for a wealth of other species. So their function is that they increase biodiversity. All-star card #2 is a strong physical forcing. Chris touched on this, Julia's touched on this, and it's a standout feature of Galapagos. Here's a temperature record from one of our study sites. It shows this 11 degrees centigrade temperature drop in 24 hours in upwelling. So this is right where we're working. It turns out that this upwelling, it's local, it's topographically influenced, and I just about fell out of my chair when I found this diagram or this SAR image of the Central Galapagos, which shows where we recorded and showed by spectral analysis that these were something called internal waves. Does everyone know what internal waves are? They're like a sine wave moving along the thermocline, and water's going down and up, and it requires a changing tide in a stratified water column. I can explain it on the board if people are interested. But look at this, right where we've recorded a steep, well, there is steep bathymetry, and we've recorded high frequency, huge variation in temperature. We see a train of internal waves impinging on our Rockus-Gordon site. If you look at the Navy atlas of internal waves, which are those SAR images and also photographs taken by astronauts, you see that they're common in Galapagos. So a common mechanism of benthic pelagic coupling. There they are impinging on the south east coast of San Cristobal. So conceptually, Galapagos is very much a pulsed environment, a pulsed temperature and pulsed resources like nutrients, chlorophyll, et cetera. Here you see a map or a chart of sea surface anomalies in the Hadley data set that our colleague Franz Smith put together. These are all places where we work, and you see the standout Galapagos with enormous temperature variability from ENSO, which is really different than the Caribbean and the Seychelles and even Palau. So I have a video of that upwelling, and it took over an hour for this talk to upload last night, so I wasn't sure if it was going to work, but it's an actual video of upwelling. And a lot of people ask us, why as benthic marine ecologists, we work on oceanographic forcing of marine communities. It's because we're particles in the ecosystem. We experience things like upwelling and downwelling directly. So let me move this along. This is a video Robbie took from one of his, we call fish cams. You notice the water is really green and turbid, and you should see it start to become really clear. And you can see those black corals, you can see density shimmering. So this is on the order of minutes. So this is what we see diving. And we've measured, sorry, I don't want that to happen. We've measured the vertical velocities. I'm trying to get to the next slide, but yeah. Okay, so, oh yeah, thanks. This slide, thank you, shows we've actually measured vertical velocities at the sites where we're doing the experiments. And this shows one of the records from Rockus-Gordon. Again, this is topographically induced upwelling, as high as 20 centimeters per second. And even higher, we've measured 60 centimeter per second upwelling currents. And you may have heard of stories of divers getting stuck. Thank you. Getting stuck in upwelling and downwelling currents, and this is what's going on. Yes, thanks. We've clawed our way up these walls in some of these strong downwelling and vice versa upwelling currents. Card number 3 is compressed biogeographic features. And this is kind of typical Galapagos, but not typical for the rest of the world. We've got 4 major currents impinging on the central Galapagos. And as Julia mentioned, we have penguins and corals coexisting. I believe this is the only place in the world that happens, as well as tropical fish. And so the boundaries between the biogeographic regions, which may be 3 or 5, depending on which paper you read, are going to shift with climate. And this has huge implications for understanding how the marine ecosystem functions. This card shows rapid biodiversity change. This is at Rockus Gordon. The number of photoquadrats versus the number of invertebrate species. 2 measures of species richness. The punchline is the number of species doubled in a year. I've studied these communities all my research life. I've never seen anything like this anywhere in the world. It's a marine community on steroids is the way I describe it, with a full complement of predators due to the high functional diversity of fish and the highest shark biomass in the world, as several authors, including Pelayo Salinas de Leon, has indicated. So this plot shows the number of species of fish versus the number of functions that are represented. So functions like a reproductive mode or a trophic category. And look at Galapagos. It's off the charts. It's the highest fish functional diversity in the world. So all this is going on. Here's another drive clogging video, but one has to show this. This is one of our study sites at Rockus Gordon. You know, huge biomass of scallop hammerheads. You see some planktivorous gringos going by. It's really exceptional. Oops, sorry, seemed to, yep, thank you. Okay, so what do? And of course, this is a biased representation of the marine ecology of the Galapagos. It's what we think's important. It's not representative of everything that's known. I didn't feel that that was my role here. And there's an excellent book on the Galapagos marine ecosystem, actually. It's the entire Eastern Tropical Pacific by Peter Glynn, who we all know of. So our approach to understanding these ecosystems is to conduct experimental manipulations to find out about the mechanisms controlling patterns of abundance and diversity. Here you see a triggerfish eating a sea urchin, a slate pencil urchin, in one of our predation experiments that showed that the triggerfish create a trophic cascade. I'll explain that in a minute. And we do the experiments alongside of long-term monitoring. So every 6 months, we go back to those sites and we do photo transects, we video record replicate 150 meter square plots for coral variation and coral bleaching. And by doing experiments in a background of long-term monitoring, we're able to understand process or mechanism. If it's a semi-realistic experiment like this one you'll see in the next slide, we uncovered the effects of top predators on this urchin-eating species. So we understand a little bit of the complexity of the system, and then we can relate our experiments, hopefully, to the longer-term variation. And so we've been doing this for 23 years, and I won't talk about all that we've done. So Yeah, that would be a very long talk. So one of the characteristics, biological, ecological characteristics, is there's really strong effects of predation. Ecologists call this top-down control. And because of the high functional diversity of fish and the presence of marine mammals, we have a lot of knock-on or indirect effects of the top predators on the middle trophic group of predators that are eating sea urchins, which are like underwater lawnmowers. And we've shown that there's something called a trophic cascade where the triggerfish reduce the abundance of the herbivores, which enables the algae to thrive. So that's a top-down effect. The bottom-up effect just means the trophic interactions are controlled by algae or nutrients. And ecologists have long argued about whether bottom-up or top-down forces are most important. And it's pretty silly because both are important. I always say no food web, but certainly predation is really important. One of Robbie's results was examining the effects of flow speed in shallow communities on foraging by reef fish. And what he found is that different species of fish are able to feed along a differing ranges of flow speed. And so parrotfish take bites at low flow speeds. Surgeonfish, excuse me, over here can feed at really high flow speeds. So it's a physically controlled guild of consumers, and they're controlled by a physical process that we can measure. So you see what I'm trying to build here is a foundation of physical biological coupling that we can examine in the future. So a characteristic of the Galapagos is we have bottom-up, top-down coupling, strong predation and herbivory, and strong bottom-up effects of upwelling, temperature and nutrients. This has been demonstrated several times in the Rocky Intertidal by Luis Fenoesa, by us in the subtitle, and by Maggie Brandt and John Bruno's group, and also in the subtitle. So it's a generalization. I just want to show one of the conceptual diagrams from the Bruno Labs research, because it's a really nice demonstration of how they analyze the effects of two aspects of upwelling, the provision of more nutrients, but also at the same time brings colder water, which slows the metabolic rate of the consumers. And they did manipulations where they showed by adding a nutrient addition treatment that the nutrient effect was only important in the warm season when nutrients were limiting. So they got more bottom-up effects varying depending on the season. So here's the way I think, or we think about ENSO conceptually. I call this the roller coaster model. And Robbie and I spent roughly a week with a photographer and writer from the New York Times who's doing an article on the Galapagos. And the only thing that was quoted was our roller coaster model. So, you know, after providing tons of input, they went out diving with us. This is what they latched on to. But I like it because it shows several things. I mean, the big question, what is the effect of multiple ENSOs that keep coming on long-lived organisms? Are they driving regime change? Or, you know, stress during El Nino, it's warm, there are negative effects on a variety of marine life, but excuse me, during La Nina, there's a resurgence of productivity. All the benthic invertebrates reproduce, diversity increases to a certain extent. But is the long-term picture running downhill due to multiple El Niños, kind of ecosystem degradation? Or is it a flat line because the positive effects of La Nina compensate for the negative effects of El Nino? So we've studied the system through 4 complete ENSO cycles. And the good news is we're seeing repeated patterns. So we're not just, doing the same thing forever. Well, we sort of are, but we're making new discoveries. I mean, a generalization that holds across 20 years is pretty remarkable, we think. So we've tracked 4 major coral bleaching events since we started in 2007, monitoring these replicate 150 meter square areas. And what you see is that 3 out of 4 of them occurred in cool periods. So cold water bleaching, in our estimation, in the Central Archipelago is way more significant than warm water bleaching. And that's not on anyone's radar screen. And it has to do with the really high temperature variability that was card number 3 in the Superstar series. So here's a visual depiction of what happened during these one of these bleaching events. We're video recording these big plots, so these are just illustrations. So here's a bleached finger coral reef at Roccaspigle in January 2008, right? As we notice the bleaching, take a look at the rock in the background. You probably can't see it, but it's the same rock. Here it is 7 months later. There's supposed to be a date coming in, I think. Yeah, July. And you can see all that coral is dead. It's been colonized by coral and algae. And here it is 11 years later. It was clearer and brighter. There's the rock in the background. There's only 1 or 2 post-leporid corals. The shallow post-leporid reef is completely gone. So this species, these groups of corals are less resilient. In contrast, the massive corals are more resilient, and I'll show evidence of that. So with Olivia Rhodes, one of my undergrad students, we looked at the effect of bleaching on the diversity of species that are associated with finger coral communities. So starting with the 2007 bleached corals, we tracked the number of species in a large number of tagged corals. And excuse me, the dead corals show the gray bars are the dead corals. The black ones are the live corals. And you see there's an elevation of diversity in the dead corals. So the skeleton was there. All these guys were really abundant. It changed composition. So there are a lot of species here. We have dead corals more than higher richness than live corals, and you see it finally dropped. So what this is an indication of extinction debt. So a disturbance happens. So it's analogous to pine bark beetles eating a conifer forest, and the trees stay there for multiple years, and the birds come in, and there are plenty of insects, so richness is high, but the functions have probably changed. So bleaching disturbance created an extinction debt of about 2.5 to 3 years post bleaching. We tracked them to a year later, and the habitat disintegrated. So here's a slide series showing the higher resilience of Parietes lobata. Robbie and I call these our iconic corals. We have a number of them that I've known since 2006. And I get bummed out when they bleach, true, but I'm happy when they recover. we have every year time series. These are just snapshots. So they chug along fine. They bleach in a cold water episode. They recover a year later, unless they're overgrown by barnacles, which boom during the La Nina year. And you can see there's a slide from January 2022 to the far right. And it's slightly bleached, but actually that coral's getting bigger. So it's doing all right. So the other effect, ecological effect we see during La Nina is a huge boom of barnacles. So you may think barnacles in the East Coast of North America, they're about this big. Barnacles in Galapagos, they're called megabalanus, and that's really a good descriptor because they grow 1.5 centimeters in diameter at an upwelling site. In a year, they're like popcorn to the food web. Everything eats them. I mean, really everything. The fish crunch them off the walls, lobsters eat them. Snail is specialized eating them. So this just shows a smaller area, 1/4 square meter over photoquadrats. You can see a lot of pink. It's crustose coral and algae. Here's exactly the same area shortly after during the La Nina. So the barnacles have reproduced all this rough appearance substrate or barnacles. So this occurs repeatedly. Here it is during the 2016 to 2017 transition event. So what we get are these barnacle roots that persist for years, we think are very important to the food web, and they may confer a type of trophic resilience by increased bottom-up effects to this really lush, functionally diverse predator community. So here is a photoquadrat time series showing the barnacle abundance trends at our 12 long-term sites. We've got data back to 1999 at 2 sites, and then we started the 12 sites in 2003. And what I hope you can see, I mean, yes, this is very messy. And these data over here are close. Well, they're not really on Eric's fantasy scale, but they're not confirmed yet because I haven't inspected them, and they're only based on 4 sites, but they're probably accurate. So what I hope to show is that there are these barnacle waves. There are these periods of about 4 to 6 years where the substrate, the bottom's totally covered with barnacles. You know, ecologically, anything higher than about 30% of the substrate covered is pretty abundant. And they seem to be associated with La Nina. I mean, we see it on the small scale quadrats. Like, look at this, the 2015 crash during El Nino and a huge boom in the 2017 La Nina. And what we found is that these patterns of abundance track with the Southern Ocean Oscillation Index with about a 6-month lag. So I can show you statistical results where the strength of the El Nino La Nina transition predicts about 58% of the variation in barnacle abundance across the whole central archipelago. So that's the repeated pattern and the physical control of the barnacle booms that we're seeing. So we also see these booms of abundance in fish, particularly planktivorous fish. So these are Robbie's data. Every time we go down every 6 months, he swims these really long transects and identifies and counts and sizes all the fish he sees. So Here we have about a 4-year record. Here's the El Nino period. And then you can see this big increase in planktivorous fish at the same time, actually, that the barnacles increased greatly. And here's some longer term data showing the density and the biomass of different trophic groups of fish. And there are kind of 2 points I want to highlight here. First of all, looking at the biomass, these are the biomass of planktivorous fish, which is right here, the yellow line. It also peaks during the 2017 La Nina, and we see a peak in the carnivore biomass in 2021, which we think, so those are sharks and jacks. We think that's correlated with the reduction of human disturbance during the anthropause. And that's a whole nother study we've done that I won't be able to touch on, but I'd be happy to talk to you about some of the results. So these are just some of the effects of the super strong 2015, 2016 El Nino. Some of these other effects, we saw a decrease in the abundance of sponges. With the warmer temperatures of El Nino, we saw increased grazing. I mean, crazy grazing by these sea urchins. This quadrat normally looks like this. It's kind of a bare geologic substrate after really intense urchin grazing during El Nino. We think they didn't have much food, so they started this destructive grazing. A big surprise, basically a lot of ecological surprises during this El Nino. We saw this bloom of cyanobacterial mats that sprang up out of nowhere, rapidly covered the substrate during the warm period and disappeared during La Nina. And the biggest surprise Robbie discovered, actually this should be 2018 if you want to find his paper, is this ulcerating skin disease in reef fish in January 2016 at the height of the warm period. And our hypothesis is that the very low food supply and the really warm temperatures stressed the fish. For some reason, this infection took off, but it was widespread, and we believe it's the first time fish disease has been recorded in the Galapagos and correlated with a climatic oscillation. Okay, so finishing up. So you see what I've been trying to do is to develop kind of physical indicators of ecological change in this absolutely remarkable, highly dynamic ecosystem. So one of the questions that we touched on in the breakout groups, and I think Jaime mentioned as well, is to focus on terrestrial marine coupling. You know, we tend to live in our ecosystem boxes. You know, you're either marine or terrestrial. Well, of course they're connected. They're highly connected in a place like Galapagos with stunning geology, with earth movements that create landslides. So here's a photograph of the south coast of Floriana. We're going between study sites. You see a relatively fresh landslide. And notice that the rocks along the water's edge are all green. So it was a recent disturbance. They're all colonized by ulva. So of course, that's a huge resource pulse. So it's a huge abundance of food. So the rubble's colonized by algae, and it extends in the rubble patch of the landslide beyond the intertidal into the subtidal. So they're all a lot of herbivores in the Galapagos, including the most charismatic endemic marine iguana that love to eat seaweed, including ulva. So that's an enormous resource pulse that extends from the intertidal that's triggered by a geological event. Furthermore, we know from Laura D's work a while ago, actually that should be 2010, that the subtidal landslide piles are an excellent refuge for sea urchins from predation. So little sea urchins have a big problem growing up in Galapagos with all these predators. So they tend to recruit and occur in high density in rubble piles. And she showed that their predation rate is, their survival is much lower. I mean, it makes sense if they're living in a protected habitat in the rubble. So, and going the other way, marine terrestrial subsidies, there are a lot in the Galapagos and they're largely unstudied. We studied the effects of sea lion pooping on plant communities at a couple of islands. And what's fascinating and well known in this ecological theory is the magnitude of the marine to terrestrial subsidy, the effect on land, is predicted by the perimeter area ratio of the islands. So that makes sense. Smaller islands have higher perimeter relative to the area. So there's going to be more influence from the edge. Also important is what these ecologists call the permeability of the habitat boundaries. So if it's cliffed, obviously less nutrient input is going to get there unless they're from seabirds versus a shallow slope. So anyway, that's a ripe area. And if it's true that those landslide rubble habitats are full of sea urchins, it could be that that represents an important source habitat to produce larvae to colonize other areas, for example, where predation is so high. And this goes along the lines with trying to understand the metapopulation connectivity of species and communities in Galapagos. Ecological jargon, a metapopulation are spatial populations, spatially discrete populations of individuals, all of the same species. They have to be connected by dispersal to be a metapopulation. A meta-community is basically the same thing, but refers to a whole group of species. So we need to understand source sink relationships in the Galapagos marine ecosystem because there's climate change. There's an increasing frequency possibly of these cold spells, cold pulses from Chris's cold pool on the west side of Galapagos. And so to understand resilience, we need to understand these connections. So our impression is that we need more studies of connectivity and dispersal that are informed by oceanography to predict passive dispersal, population genetics, and the spatial patchiness. I just want to highlight a colleague's research who is taking this approach with the sea turtles that are so abundant, fortunately, in the Galapagos. He's using, this is Franz Smith, and in conjunction with the park, he's using drones to map out the high density patches of sea turtles and where their most important nesting beaches are. So he can really understand basically where the key source habitats are. Another suggestion coming from our work, and we've done this a little bit, but not very much, is that we should look at ENSO as a treatment. It's a huge ecological climactic force that regularly impacts the Galapagos, as we all know. So to understand what's really happening in terms of ecosystem functioning, we should conduct simple-minded experiments through a series of El Niños, La Niñas, and see how ecological processes change. And the problem with El Niño and so ecological research is it's mostly impact driven. You know, I've been fortunate to get rapid grants from NSF, you know, because there's this huge El Nino hitting and La Nina resurgence. But if we knew, you know, we know when they're coming, if we could design these simple-minded experiments to inform how ENSO and or El Nino and La Nina affect ecosystem functioning, then we'd really be at a kind of higher level of understanding. I'll show you a slide of what I mean in a minute. I pitched this idea in 2016 at the SAMAR conference on climate change in the tropical Eastern Pacific, but so far no one's picked up on it because it is a lot of work, but a network like this, it would be a great coordination. So basically, we could deploy caged and uncaged colonization plates. We'd get algal biomass during El Nino, during La Nina. For the exposed plates, we'd get the consumption of that algal biomass. So that's ecosystem functioning. If we put GoPro cameras on them, we'd record the biodiversity of consumers and how that varies through El Nino and cycles, and we'd get the effect of biodiversity on ecosystem functioning. Okay, I think this is my last slide. So it's an argument for taking more of a vertical perspective to ecological and climatological research in Galapagos. You know, we know the water column's stratified. We know there's strong, very strong, I hope I've convinced you, benthic pelagic coupling. All these ecological processes vary along depth gradients very strongly. Yet in ecology, at least, we're only looking at one depth often. There's some exciting deep sea research that's being spearheaded by the Charles Darwin Foundation that's really taking a large depth perspective. But for example, are the cool deep communities cool in many ways a depth refuge from warming, from bleaching? How does temperature stress affect the physiology? And that varies with depth. John Bruno's lab at the Galapagos Science Center is doing some very exciting research about that. Ravi and I have long thought about how fluctuations in the thermocline affect predation and herbivory. Again, a vertical, possibly climate control of an ecological process. How does the zonation, the depth zonation of foundation species like black corals vary with depth? And, you know, what is their ecosystem function? Does that vary with depth? For example, their biodiversity enhancing function of the foundation species. And just underwater alone, are there subsidies like elsewhere in the world, like in the Amazon, where logs float offshore from the river and sink to the deep sea and represent a resource pulse? As far as I know, you know, this type of shallow to deep ecosystem exchange is almost entirely unknown in the Galapagos. And my last pitch is to think about scaling between very large scale oceanographic forcing and smaller scale oceanographic forcing. So relating kind of the basin scale oceanography to mesoscale and local scale oceanography, where ecologists are actually doing experiments. And this has been my pet peeve forever. Sorry to plug it again, but for example, in our studying of upwelling ecology across those 12 sites, we started out by actually measuring the strength of upwelling in front of these walls where we're doing experiments with barnacle predation, and documented a gradient of very strong upwelling. So there's often a mismatch in ecology, marine ecology, between the scale of climatological oceanographic knowledge and what's really happening at the local community. My last content slide is this one. It seems we've forgotten about evolutionary processes underwater. You know, I teach this stuff at Brown. I start off my ecology course emphasizing that evolution is occurring on the order of decades. And, you know, here we are, here's our study site at Daphne Menor. We've been working there for 20 years. We've never done, I'm sorry, a single evolutionary investigation. Yet right next door, Peter and Rosemary Grant are sitting on Daphne Menor and, you know, documenting the effect of El Nino as a natural selective force driving changes in beak size. And I've talked to Peter and Rosemary, and they say, yeah, we look at your island all the time, or when they used to go out there, and we wonder what's going on, and we like, you know, what you found. But yeah, they didn't give me this idea. But for example, are corals evolving to these multiple, to adapt to these multiple disturbances from repeated El Ninos? Quite possibly. Okay, so that's it. Thanks for your attention. I've got one acknowledgement slide. Sorry for the technical inabilities. Thanks to the Charles Darwin Foundation, which is our home in Galapagos, and to Maria Jose, who's the science coordinator, Marta, who arranges our logistics, Galapagos National Park and its director, Danny Rueda, who allows us to do this, National Science Foundation for funding, various NGOs who've helped in a pinch to keep the long-term work going, the Nature Conservancy, Galapagos Conservancy, and Conservation International. A big shout out to the captains of 3 different vessels who put up with us in very long days, the Mabel, Valeska, and Pirata. We spend a lot of time underwater, and, you know, we use every hour of daylight. And, you know, these are just some of the students, graduate and undergraduate, and colleagues who've worked with us over the years. And of course, this is a huge team effort. Particularly, I'd like to acknowledge Maggie Brandt, who did a PhD with us and is now a professor at Universidad San Francisco de Quito, and Franz Smith, our long-term collaborator, who's is having his photograph taken. So.
All right.
So sorry, everyone.
All right. So thank you, John, very much.